today. Hello, good morning, everyone. So it's with great pleasure I would like to introduce today's guest lecturer, Professor Carola Niesler. Professor Niesler is an Associate Professor of Biochemistry in the School of Agriculture Sciences, University of KwaZulu-Natal, South Africa. She received her PhD from Department of Clinical Biochemistry, University of Cambridge in UK, and has more than 25 years of experience in the research area of stem cells and application of their cellular products in the broader context of regenerative medicine. As an established scientist, she has graduated more than 60 postgraduate students and published more than 40 internationally peer-reviewed articles that relate to regeneration in health and disease. So today she will talk to us on the title Extracellular Vesicles, Having Fun with the Nature's Nanocarriers. And Professor Niesler, the floor is yours.
Great, thank you very much. I'm going to share my screen. You just let me know whether it's sharing and whether it's on slideshow. Let's see. Can you guys see it?
It's not in the slide. Now we can see it properly.
Can you see it? Okay, let me just move a couple of things around here so that I can see what I'm doing.
Yeah.
Okay, great. Thank you very much. Thank you so much to yourself and to Professor Fazeli for allowing me to present on this platform. Yeah, I think I'll just get going. As you've mentioned, the title of my presentation is Extracellular Vesicles, Having Fun with Nature's Nanocarriers. I'm based at the University of KwaZulu-Natal. That's in the province of KZN, KwaZulu-Natal, which is up on the east coast of South Africa. So where the water is warm and nature is beautiful. We have 5 campuses, and we're on the Pietermaritzburg campus, which is slightly inland, and we're surrounded by really amazing nature. So we're very lucky to be located in this particular part of the world. Let's see if I can get this to move. There we go. I wanted to start the presentation off with this quote by Einstein, which is, "I have no special talent, I'm only passionately curious." So this is related to the fact that, I entered the scene of extracellular vesicles a lot later than probably most people listening to this presentation. And it's because I just became curious about what are these EVs and what do they do and what can we know about them and how could we use them. But it also applies to the rest of my research. Most of what I've done in my life has been driven by just curiosity in terms of the areas that I've worked in. So maybe just to give a little bit of a background, I was born in South Africa and I did my undergrad at Stellenbosch University. But as you mentioned in the intro, I was lucky enough to be able to do my PhD in Cambridge, and there I was working on pre-adipocytes, so the cells that we isolated from the stromal vascular fraction. And at that time, which is in the sort of mid-90s, the whole field of stem cells was really sort of starting to grow and expand and become sort of the big thing. And at that point, when we were working on these pre-adipocytes, we referred to them as pre-adipocytes and never really adipose-derived stem cells. And really, in hindsight, realized what we'd been working with the whole time was ADSCs. And we were looking at apoptosis of pre-adipocytes in the context of diabetes and obesity. So I'm really trying to figure out whether pre-adipocytes undergo apoptosis, which at that point wasn't really known. And it's weird to think that now because now we know a lot more. And whether that could be used in any way as a way to control weight in an individual. From there, I moved to the University of Manchester. So went further north and I did a postdoc for a couple of years up there. Not really working on stem cells as much, but moving more into wound repair and regeneration and looking at cutaneous wound repair. And I worked with a group that was largely funded by companies like J&J and Smith & Nephew. And we were particularly focusing on TGF-beta 1, 2, and 3. Specifically looking at the fact that TGF-beta 3 is known to be anti-scarring and TGF-beta 1 and 2 more pro-scarring. And so really trying to understand the juxtaposition between those different isoforms in the context of wound repair and regeneration. So then in 2001, I was insanely homesick, so I returned to South Africa and returned back to Stellenbosch, which is down at the bottom here of South Africa in the Western Cape. And I was inspired by a paper that appeared in Nature, which was published by the group of Ulrike and Versa. And they had looked at bone marrow-derived stem cells and the ability of these stem cells to promote the repair of the heart. And so when I started at Stellenbosch as a young lecturer, I started working and establishing a group that would look at stem cells in cardiac repair. And then I expanded that into skeletal muscle repair and really tried to get to grips with that whole area of research. Around 2006, I moved up to KZN and the University of KwaZulu-Natal and continued to work on skeletal muscle stem cells, which we call satellite cells, but focused more then on modulators of their activity, their proliferation, their migration, their differentiation, and looking there at all the factors that are in the extracellular environment, whether that is your extracellular matrix factors, your growth factors, other cells, and how all of these affect the whole process of proliferation, differentiation, migration, etc. It was around 2009 that I became familiar with NEXT Biosciences, which had been launched in 2005, so it was still a young cord blood banking company. And I became familiar with them, introduced to them, and then began as their scientific advisor, if you will. And they've just celebrated their 20-year anniversary and are a lot more broader now in terms of their remit. They look at a range of products for wound repair. They do genetic testing, and then they also still do stem cell banking and the culture of adult stem cells as well. So in a nutshell, my background really is mesenchymal stem cells, the understanding of their proliferation, their migration, their differentiation into a range of somatic cells, for instance, myotubes and myoblasts, how other cells and factors in the extracellular environment affect the ability of stem cells to become their destination cells, and how this plays a role in wound repair and regeneration and also in disease. And it was around 2018 or 2017 that I was having a discussion with a colleague and a collaborator and a friend down at Stellenbosch University, Professor Myberg, and she had begun working on extracellular vesicles. And she was particularly looking at microRNA content in these EVs and trying to understand how this is important for the response of the body to muscle damage following bouts of exercise. And so I started to wonder whether extracellular vesicles might be responsible for the delivery and the mechanism whereby these factors that we've been working on are delivered to cells and are actually modulating the response process. So that was the start of our EV journey. So I think I'm relatively young when it comes to EV research, relatively inexperienced in comparison to many others. And we've had quite a sharp trajectory in terms of the learning curve. And if you look at the history of EV research, we've really come into the fray here around 2018. And so there's a long history here that we can learn from, which is a real advantage because we can learn from others a lot quicker. We don't make the same mistakes that everybody else has made because we've come in relatively late. But the disadvantage, of course, is that we have a lot to learn and we have less time. So, of course, we know that EVs in the 1900s, when they were first sort of identified, I guess, were thought to be the sort of the carriers of trash, right? That was what their function was thought to be. And it was then, over time that it was realized that actually these are really nifty communication tools. They can take information from one cell to another, and they can protect the information that is associated with those EVs. And so the first trial using The dendritic produced EVs was back 20 years ago in 2005. We now have MySafe guidelines that have been optimized and allow us to standardize our research more. And we have a lot of EV-centric trials on the way. We also know that EVs, there are many different types of EVs, many more than we thought of 10 or 15 years ago. So when I started in the whole area of EVs, we really characterized them as either exosomes or microvesicles or apoptotic bodies. It was that sort of those three. And we now know over time that, you know, there's a lot more to it than just those. And there are even these smaller exomeres and supermeres. But the common denominator is that these EVs are able to take this cargo, whether that cargo is inside and it's a microRNA or it's an enzyme or it's a protein or whatever it is, or it could be in the membrane, could be a lipid, could be a receptor, or it could even be on the outside here, and the outside of the corona chemokine, can take this and it can ferry it from one group of cells to another. And it does it in a way that is seemingly more efficient than if that particular growth factor, for example, was just swimming around in the vascular system. So when it gets to its site, it can then bind or fuse or interact in some way with a target cell and influence various aspects from gene expression, protein expression, mRNA degradation, etc. So we know all of this to a certain extent at this point, and that's to me what makes these EVs so fascinating to work with. So that brings me to the start of our EV research, which, as I said, was around 2018 and was in connection and collaboration with NEXT Bio. So NEXT Bio has a number of products that utilize various products of the placenta for the purposes of wound repair. So that includes, for instance, amniotic matrix in the form of amniomatrix and amniorederm, but also cord blood serum in the form of a product called OptiSerum. And they were interested to know which are the factors that, or which factors are found in this cord blood serum, that in the product that they have. And this was based on the fact that there was already information telling us that cord blood serum is really enriched with a range of pro-regenerative factors, and it has more of these pro-regenerative factors than the adult autologous serum. And so some of these are shown here: vascular endothelial growth factor, hepatocyte growth factor, which play a role in angiogenesis and tissue regeneration, TGF-beta, which we know is important, particularly TGF-beta 3, Grow alpha, involved in wound healing and the inflammatory process, and others. And so we were interested to know, or NEXT Bio was interested to know, well, you know, are these factors in the product that they are selling? So OptiSerum is a commercial biological eye drop product, which is shown here on the right. It contains 20% umbilical cord blood serum, and the rest is saline, and it doesn't have any preservatives in it. And it's used to promote ocular repair post-surgery in all corneal, when an individual has things like corneal abrasions. And the basis for this product is there were a number of publications which showed in other labs that patients with corneal defects or keratitis or severe dry eye syndrome can show high rates of repair or healing when treated with cord blood serum rather than adult serum. And that probably relates back to the fact that the cord blood serum has more regenerative factors in it than the adult serum. So probably related to these regenerative factors. So the first question was, okay, so in this product that we have, what is our protein complement in there? And so we got 14 batches of OptiSerum, and we analyzed those using a 48-plex. And so what you can see here is just the different protein levels that we could identify and could measure. So you've got GROW-A there, and this is in picogram per mole. You've got HGF, VEGF, and then you've got others which are really very, very high, your stem cell growth factor B, your PDGF, MIF, etc. We also carried out an ELISA because TGF-beta3 wasn't on this particular panel, and we could detect a few hundred, 800 to 900 picogram per mole in our samples. And at the time, a publication from Buzi also showed they'd done a similar thing, and our levels were actually very similar to what they'd shown, which was really cool. If you look at TGF-beta3, they were picking up just over 600, and we were picking up 870. So similar region. Let's have a look at PDGF. We found slightly higher, but again, in the thousands when it comes to the picogram per mole, and VEGF as well, slightly higher, but similar region. So that gave us quite a lot of confidence that we had, or NEXT Bio has a product which is enriched with these pro-regenerative factors. We did some additional work where we combined these the cord blood serum together with the amniotic membranes, and we had a look at the ability of this combination to promote wound repair in diabetic wounds using a mouse model, just the OB-OB, which has a particular, in this case, in the lab of Professor van der Vyver, the wound is treated in such a way so that it doesn't close over the 14 days. And so what we found was the combination of this cord serum and the amniotic membrane actually really had a very beneficial effect on the underlying granulation tissue and the remodeling that takes place, and was able to promote wound repair. But what about EV? So that really was the final interest here when it comes to the cord blood serum. So we confirmed that the serum has a lot of pro-regenerative factors in it, but are these in the extracellular vesicles? And to look at that, we first had to see, well, can we isolate the EVs from the serum? And there had been some studies on EVs from cord blood, from the plasma, for instance, from stem cells that were isolated from cord blood and then allowed to express release the EVs. Some of these studies are listed here. So in 2018, there are a couple of studies that looked at, for instance, cell proliferation, endothelial cell proliferation and tube formation, or fibroblast cell function, and they were found to have a positive effect, the EVs. A little bit later, the EVs from the cord serum were shown to have potentially an immunosuppressive function, and then more recently, potentially a neuroprotective effect, and this is in a Parkinson mouse model. But still very, very little out there on EVs from cord blood in general when compared to EVs from other sources. And also very little known about the protein cargo, which is what we were primarily interested in. So from those OptiSerum samples where we'd analyzed the regenerative profile when it comes to the proteins, we isolated the EVs from those paired samples. And we used a very basic PEG 6000 approach. We wanted to get the biggest yield so that we had enough EVs to work with and we could use those in a multiplex system. So this was actually my first master's student on extracellular vesicles, Spem Kieser. And so we were floundering around in the dark a little bit. We really didn't know initially what exactly we were looking for. So it took us quite a while to really figure out what EVs look like under transmission electron microscopy. And now we can do that regularly. And with all the EVs we work with, we tend to find these beautiful cup-shaped morphologies. We were using the tunable resistive pulse sensing. So that's the Xoid from Izon, which uses a nanopore to measure the number of vesicles and their size. And so, every time a particle moves through the pore, there's a change in the current, and that can be used following calibration to determine how many vesicles you have, what their sizes are. And if you increase the pressure at which you're pushing these vesicles through, you can increase the number of vesicles that you analyze. So we utilize this technique probably for about 4 to 5 years to characterize and to determine the concentration of our EVs. So it's a nice technique in that it's very accurate, but it's a very hard technique to use on a consistent basis. So when you're looking at big sample numbers. But we did find that our EVs were in the right size range. We did some very basic flow cytometry together with NEXT Bio on their DX Flex to just look at CD81 and CD9 expression, and we did detect CD9 and CD81 either separately or together. And I call this very basic flow cytometry because we weren't using any beads at this point. We really were learning on the trot, but it was encouraging to us that we were picking up this type of signal. We carried out a couple of cell culture experiments to just have a look and see whether our EVs have an effect on the growth of different cell types. So these are your mouse macrophages, and these are the human HEC293 cells. And when you add anywhere between 10 to 100 million EVs, you're getting a nice significant increase in the cell number. So that tells us that these EVs have proteins associated with them, either inside in the with the membrane or outside, that is being delivered in some way and promoting this effect. One thing that we didn't do here, which we need to go back and do again, is we'd like to take that serum and actually deplete it of the EVs and then add it to the cells and see how does that effect compare with the EV and with the serum which has not been depleted, so that we get an idea of the real function and the effect that these EVs have on the cell growth. So then we looked at the proteins that were associated with these EVs. So we had 48 in our 48-plex. Of the 48, 36 proteins had been detected in the cord blood serum itself. So there were 12 that we couldn't detect at all in those earlier studies. And of these 36 proteins, 22 were found associated in the EV fraction. So that means that 14 were not found in the EV fraction, but were found in the cord blood serum. And those that were in the EV fraction, those 22 are listed here. And many of them, like Rho alpha, HGF, VEGF is in there somewhere, are known to be associated with regeneration. So this was really interesting to us. What was quite curious was that in some cases, for instance, IL-6 and VEGF, which I've indicated in red, now these two were actually not detectable in quite a few of the cord blood serum samples. So that means in the original cord blood serum, we couldn't actually detect VEGF or IL-6 in some of those samples, but we could detect them in the EVs. So that for me is quite curious, and I'm not sure what the reason for that is, whether that does reflect that there's a production that is actually enriching these factors in the EVs, or whether this is to do with these factors are more sticky and sticking to the outside, or they trafficked into or enriched into the EVs from the outside, or how it actually works, we're not sure, but this was actually quite curious. So what we then did is we tried to standardize, and this was actually in response to the conference last year in Austria. We tried to standardize the level of proteins that we had in these EVs. And so what we decided to do was to express the level of proteins per 10 billion EVs as a way of trying to understand what is the association of the different proteins with groups of EVs, so per 10 billion in this case. And so you have some like macrophage colony stimulating factor and MCP3 and eotaxin, which are really very low. We're sitting at 10 picograms per 10 billion EVs, so that really is very low. You have others like stem cell growth factor B, which are sitting at a far higher level, and then the rest are sort of in between. To make sure that what we're measuring there is indeed accurate, we just took one of the factors that was expressed at a reasonable level, which was IL-15, and we did an ELISA on that. And we compared the ELISA data to multiplex, and we could see that the levels of IL-15 that were detected using the ELISA were similar to that detected in the multiplex. So that was confirmation to us that the data that we're getting here is indeed quite reproducible and quite accurate. So we then wanted to try and understand a little bit about comparative levels. So not looking at the actual levels, but looking at comparing the levels in the EV fraction versus the original serum. Now, what we didn't do here, and what in hindsight and retrospectively would have been really good, would have been to take that serum fraction and to take the EVs out of that. And then to measure what was left, of course, so that you'd have data on the EVs and you can have data on the serum minus the EVs and then the serum plus the EVs and see whether it all matches up. So we didn't do that, and that was something we can look at doing for certain factors going forward. But if we look at that comparatively, then what we can see is that, for instance, NGF, which is expressed at very low level. So you can see where is an NGF on here, there. So it's sitting at about, it's at about 100 picograms per 10 billion EVs. And even here, if we go back, the levels of this is there is very, very low in the serum itself. So what's interesting is it seems to be concentrated much more in the EV fraction. And if we consider that this little white sliver here includes serum that actually has EVs in it as well, because we didn't deplete the EVs, then that makes the fraction that is free in the serum even less. So that's quite interesting. So I would potentially suggest that EVs are enriched for this particular factor, I think, but I stand to be corrected. Similarly, VEGF, which, if you remember, VEGF detection in the in the serum component was quite variable. There were some instances where we couldn't actually detect the VEGF, so it was incredibly variable, whereas we could detect it in all the EVs. So is it the case that maybe VEGF is enriched more in EVs in cord blood? That could be a possibility. The other growth factors, even for instance, stem cell growth factor B is detected at high levels. It's still, when you compare the levels to the serum, it's less in the EVs as for the rest of these. So in this case, these ones I would say are, I would assume are then therefore less enriched, whereas the ones in A are more enriched. What does that actually mean? I'm not entirely sure, and we need to do some additional experiments and think about sort of how we would approach that to try and figure out what's the role of all of this. We did the same then for cytokines and chemokines. So if we look at the IL-15 and TNF alpha, could potentially be more enriched in the EVs compared to the serum, whereas the rest are actually much more prevalent in the serum itself and less so in the EVs. And then if we look at the chemokines, what was quite interesting was GROW alpha. So this is one that seems to be a little bit more concentrated in the EVs when compared to the serum. And this is, of course, one of the factors that is incredibly pro-regenerative, whereas the rest are actually quite minimal. So I've put a question mark there because what does all of this mean? I'm not entirely sure. This is something that we're now working on to try and understand. Does it reflect enrichment? Does it reflect better protein delivery? Is there a role there for regeneration? Is there a role in development? Because, of course, cord blood serum is found in the umbilical cord, blood, and that's what connects the mother to the baby. And so is there a reason for these factors to be maybe more enriched or less? And does it have a role to play during development in the communication between the mother and the child? And this was actually when Professor Fazeli gave his talk last year. This was something that really interested me. His talk was really fascinating and looking really at that communication, and got me thinking really about EVs and their role in communicating back and forth between the mother and the child, and whether they have a role to play in contributing to the delivery of not only good things, but also bad things, right? So things that could damage or could delay development or could influence the child later once it's born. So this was a discussion we had then at the conference, and I'd love to, when I have a little bit more time, take it further. One thing I really do worry about, though, and I'm not sure if my worry is unfounded, is the purity of our samples. And I know PEG is not the ideal way of isolating pure EVs, that we are acutely aware of. When we look at our EV yield from the cord blood serum, it's fine. We're getting 25, 30 billion per mol of cord blood serum. But when we're looking at the purity, the particles per microgram protein, we really are not in a very pure space there, and that's not ideal. And that, I think, is probably down to the contaminating lipoproteins, and may reflect the fact that we need to use different isolation methods depending on the source of our EVs. And I say that because when we look at conditioned media, so this is media from 3T3L1, pre-adipocytes or cells that has been generated by one of my master students, Tatum Thomas. And when we look at the purity there, and these are EVs that are secreted into a serum-free environment, we are sitting in a much better purity, and we're using PEG to isolate this, so the same approach as we used here. So maybe we need a different isolation method for serum compared to conditioned media. And so this is something that we've started to look at. We've looked at a range of different isolation methods and whether that influences the purity of our EVs depending on the sample that we're working with. So a study that came out a while back did actually confirm that when you use PEG, you should be able to reach a 10 to the 9, right? So this is entering the pure zone. It's not very pure. Yeah, 10 to the 10 and 10 to the 11 is published as being really, really very pure. But just using, anywhere between 5 and 8% PEG, you should be able to reach a reasonable purity. So again, I think we need to adjust our method based on the source of our EVs potentially. And so that needs to be a bit more bespoke than we originally thought. So this is sort of the end of the cord blood serum EV section. So what does all of this mean? So there's a lot of more work to be done. We've learned a lot. Straight roads do not make skillful drivers, so sometimes we've gone around in circles again and again and again, but I think that has helped us learn from the mistakes we've made and to make better decisions going forward, and hopefully that means that the progress we make now will be faster. So this conference, which I've referred to, which was last year and was the first international EV conference I was able to attend, really was hugely impactful for me. I met a lot of amazing individuals, including Professor Fazeli, and listened to a lot of amazing talks and learned a ton from this. One group that I met and person that I met was Rosa from Particle Metrics. And after the conference, we were then able to source funding to actually get our own Zeta view, which has made a huge difference in our research because we can now process samples a lot faster. We can also do some really cool fluorescence studies. We can use the tetraspanin kit, which we're starting to optimize. And so the amount of output we can generate and analyze has just sped up, and it's allowed us to really start to collaborate a lot more with many people on a lot more types of EVs, whether that be bacterial EVs or plant EVs or EVs from serum or saliva, or maybe, as we were talking before this presentation, from rhino follicular fluid. All of it we're insanely interested in working with because EVs are just fascinating. So currently we are looking at additional purification steps. We're trying to characterize our cord blood serum EVs more accurately. We've used the cell mask orange dye. We're busy optimizing the tetraspanin detection kit and carrying out a couple more ELISAs for confirmation of levels and doing some cellular assays. But what would be really quite cool is we have a newly established zebrafish research facility, which we established probably about 3 years ago, and we're really keen to establish an in vivo wound healing model in those zebrafish, and then use some of these EVs to understand the effect of them on in vivo wound healing in the zebrafish model, and then try and modify maybe and try and figure out which of those factors that we mentioned that may be enriched, which of those are actually important. Can we generate EVs which have high levels of only NGF, for instance, or only VEGF, and add those to our wound model and see what the effect would be? So all of this is stuff that we're sort of looking at the moment. So I wanted to just change direction for the last sort of 5 minutes. So more recently, we've started to, and this is a direct function of being able to acquire the Zeta View, we've started to look at other EVs, and I just want to touch on 2 particular areas. The first is salivary extracellular vesicles, and then the second is my foray into plant-derived EVs. And I'm going to focus on the cannabis work that we're doing there. So again, this is all about curiosity. It's especially the plant-derived was purely curiosity-driven. Do these things have EVs? What do they look like? Can we characterize them? What does it mean? How can we use them to have an impact? So if I start with the salivary EVs, I came into this as a collaborative co-work, I would say. I have colleagues, Professor Makraj and Valji, and also a postdoc, Dr. Ibrahim, and PhD student, Cornelia, who have a lot more experience in sepsis and circulating exosomes in sepsis as diagnostic biomarkers. So they've published a couple of papers on this. Why sepsis? Well, sepsis has a very high mortality and morbidity. This is highly related to the fact that delayed and inaccurate diagnosis means that by the time you realize the patient is septic, it's almost too late. And certainly in South Africa, we have a very high mortality in ICU and a very worrying mortality hospital-wide. So the idea is, can we find ways in which we can diagnose the sepsis earlier and in a less invasive manner? And so EVs would be ideal for that. EVs from serum are one aspect, but EVs from saliva are even less invasive. So there came our first foray into saliva EVs. Can we isolate them? What do they look like? So Cornelia has been doing some optimization, and we can isolate these EVs, and they have the lovely sort of shape that we're looking for. We've run them using a variety of isolation techniques on our Zeta View, and we're getting reasonable concentrations, around 50 billion per mole. That's a reasonably pure population. 80% of them are under 200 nanometers. And so once we've optimized this, the next step is we're looking at protein content, and then we're going to look at nucleic acid and purity. We then want to do some chemokine or maybe multiplex or ELISA assays on these, similar to what we did with the cord blood serum, but maybe learning from the mistakes we made there. If we can, we'd love to do nanostring if we have enough funding. Otherwise, we'll look at some qPCR to look at the microRNA profile. And very importantly, we want to look at the tetraspanin expression and colocalization. That's based on, if we go back, a paper that the group published last year, which looked at the tetraspanin profile and found some really interesting differences in control versus septic patients. For the plant-derived EVs, this is a very steep learning curve. I thought it would be quite a simple area, but it's clearly not. I initially thought plant-derived EVs were all you just call them plant-derived EVs, but I realize now that basically if you're culturing your plants, then you can call them plant-derived EVs or plant extracellular vesicles. But if you're isolating them following homogenization, you probably should rather call them plant-derived nanovesicles, because there's a lot of other things in there. So we're really looking at plant-derived nanovesicles, and this is a project that a master's student, Thando Situle, is busy with. She's isolated these EVs from cannabis sativa, so specifically Swazi Gold. We have a collaborator in horticulture who has a license to grow and to isolate from the cannabis, and we're working intensively with him on this. We can isolate a fair number of EVs. They are actually quite pure in terms of their diameter. And we've done some initial HPLC. This is not the EVs. This is just our standards, right? So if this were the EVs, I'd be jumping up for joy, but, well, maybe not. But So this is just our standards at the moment, and what we're now busy with is sending our cannabis EVs through the HPLC to see, well, what is the CBD content, the CBG content, and obviously we would prefer that to be higher than, for instance, the THC. We would expect it to be higher because this is cannabis sativa, which is supposed to have more CBD in it than THC, but we're not sure. Thunder's also running a couple of wound healing assays, just in vitro wound healing assays to see what kind of effect these EVs have in vitro. So I think that takes me to the 40, sorry, I'm 2 minutes over. So just to sort of summarize, through our umbilical cord blood serum work, we learned a lot about the regenerative factors that are in serum and about the EVs that are in serum and the fact that they may possibly be enriched with certain proteins. We're unclear whether they are involved in delivering the pro-regenerative effect of these of the serum on in the eye, for instance, or even in a wound environment like we showed in the diabetic mouse model. So we need to do more characterization, and we may need to actually generate better purity when it comes to these particular EVs, and we're looking at that. For the saliva and the cannabis, this is very new work. We've also worked on citrus EVs, and we're collaborating with a group in biochemistry that's looking at the bacterial EVs, and I didn't present her work because that is maybe something for another presentation that Dr. Mkhize can present on. But we've established the protocols to isolate these EVs, and we're now in the process of characterizing their protein, the nucleic acids, hopefully microRNA, and the cannabinoids. But we are insanely keen to collaborate with like-minded individuals who are excited by EVs and want to know more about them, because I think that's the only way you move forward is when you work in teams and you complement one another. So with that, I'd like to say thank you very much for this invitation. They always say there's no I in teams. I love working with people, and these are some of the people I work with. They're all absolutely awesome. Students we have are awesome. The academics are awesome. The industry partners are awesome. And it's just been an absolutely amazing time the last 8 years working on EVs, and I'll take any questions. Whoops, those are my cheat slides.
Thank you very much, Professor Niesler, for the interesting lecture on EVs from very interesting sources. Now the floor is open for questions. Until others come up with questions, I have a question. What do you think are the potential application of EVs from cannabis?
Yeah, well, the CBD and CBG are known to be in pro-regenerative when it comes to wound repair. There are a couple of studies that have shown that they are known to have antioxidant activities. So one of my thoughts was, is this something that could be used in a wound healing environment, sort of part of the cream? Maybe could you have EVs from cannabis that are in creams? I'm sure people have already done some of this somewhere in the world that I haven't read of, but could you deliver your cannabis, your CBD, for instance, more specifically without the sort of more psychoactive contributors? If you isolate your EVs from particular strains, so could we compare the EVs in terms of their components from different strains and see whether there's certain have EVs that have more CBD or CBG? And it's a very simplistic approach, but maybe this is one way of delivering the good stuff that is more pro-regenerative, and see whether that would be a way to have an effect that's more targeted. So that was sort of the idea behind that as an application in the future. Yeah.
Yeah, very interesting. And also when you talk about umbilical cord blood EVs, and you earlier mentioned that you already tested umbilical cord blood serum on diabetic wounds and it had an effect. And did you try with your EVs at least one trial also? No,
not yet. So that that's what we're going to start with. So So Professor van der Vyver is down at Stellenbosch University, and we are hopefully maybe end of the year, beginning of next year, going to start with that. So we just want to make sure that the way we structure it, that we learn from mistakes we've made in the past, and we structure it in the way that we have the different components, and we can really say exactly what it is that's having the effect. So yeah, what was really interesting about that study was that there was a particular effect on the remodeling. So one of the things in diabetic wounds is, they don't close very efficiently. So they become what we call hard to heal wounds. And that means that over a 12-week period, you're actually not able to heal these diabetic ulcers. And also, if you heal or manage to heal them, the chance of them opening again is actually a lot higher. So that re-injury. And what we saw with that particular study using the amniomatrix and the serum was the remodeling was a lot stronger than in the controls. And so that was quite interesting, because it would suggest that the underlying repair, so you're not just getting reepithelialization, you're actually getting good remodeling. The fibroblasts are working hard to put that remodeling and the collagen down effectively below the surface. So yes, let's see whether the EVs have the same effect.
Yeah. Let's see. Yeah, please, Alias.
Yeah, thank you very much. It was very, very interesting. I think regarding your saliva work, you would definitely have more, how can I say, easier life, or I hope at least, compared to the serum with the infants, because it's a less busy material. At the start of the material, you would have simpler, and probably I'm actually thinking that's a very nice one, the diagnostics, and then seeing that if there would be a difference between the somehow patients that would undergo sepsis and those that they don't, and probably it can be a very nice diagnostic tool in the hand of the clinician.
Yeah.
But like us, we are also constantly thinking, how can we bring our EV research to the field, to the application? And that is also the reason we have also started to work with plants, with the different parts. We find out it is, or we find out it is very, very difficult if we want to bring our work that we do with the embryo and the others into the application. But that has been why we have gone towards the other sources of EV, and also making the EVs is also, very expensive when it comes from the mammalian tissue also.
Yep. I concur totally. I mean, I think it's exactly the same. It's working with plants has been so interesting. It's much easier, and so you can do a lot more. But it's also just fascinating to realize how much communication is going on in plants. So yeah, concur completely.
The one other aspect area that here we are somewhat trying to get involved, and you being in geographically where you are is also very much possible, is we are very interested in Onehealth, in the effect that the approach of having human, animals, environment in one point. Do you think there is any potential, as you come from South Africa with all that nature, animals, everything that is there, and environment, and so do you see any potential of EV application in Onehealth approach in there? Is there anything that you think there would be a possibility of collaboration, cooperation, or interaction in that respect?
No, definitely. I think when you refer to it in the context of Onehealth, you are talking specifically about the overriding or the synergies between, for instance, extracellular vesicles in the human world, in the animal world, in the plant world, and how we can move between kingdoms basically and apply the EVs, right?
Potentially that, but also for the issues with the antibiotic resistance, or there are quite also a lot of other points, or maybe the role of the environment in the whole thing. And so just It is something I thought that maybe you would have, because being in South Africa, maybe you are more exposed to this compared to us here in Estonia. I wanted to know if you had any experience on that regard.
Yes, so I'll share two examples maybe. The one was I've worked with a colleague in micro, and we were looking at fungal EVs, and trying to see whether they would promote the effect of various products they were looking at in an antibiotic type setting, in an antibacterial setting. So that is one aspect, but I think maybe of more interest is I'm working with another colleague in genetics who works a lot on eDNA, so environmental DNA, and so we're really interested to see whether we can look at different wildlife, and we're looking at things like crows and vultures, and see what can we learn from the tissue of these of these animals by extracting the EVs, but also particles, so plastic particles as well, and what does that tell us about the health of the environment? So I think EVs are ideal, or nanoparticles actually, with EVs being part of that, are ideal as a way of linking your environmental and your human health and the effect that they have on each other. So those would be the two areas that we are particularly interested in: your wildlife species, the isolating and characterizing the extracellular vesicles in the animals in the different environments, linking that maybe to eDNA. How would we do that? Because eDNA is a great way of figuring out what's been in an environment. So yeah, those would be the ways. So I'm incredibly keen on working across different disciplines, because that I think is incredibly exciting, but also the application is huge. And I agree with you in terms of when you're looking at mammalian EVs, so you're generating EVs from cells, so we generate them as you do, I'm sure, as well, from the variety of cell types in vitro. But it's expensive, and it's a lot of work. And isolating them from plants is a lot easier and may have a better impact. Isolating them from animals to understand the health of the animals and how that could impact on humans, that could be really impactful, because I think that's very important given the state that we find our environment in. So yeah, I'd be very keen to just talk to people and understand how what we work with here, what we have access to, how that can fit into a One Health approach. Yeah.
Thank you very much. Yeah, and hopefully, yeah, we would find a way to collaborate, cooperate. Then maybe we can reach to South Africa and see all that beautiful environment you have. You are not going to come to MOVE meeting here in Estonia.
No, I really wanted to, but I have not been able to. There's various challenges. So maybe the next meeting could actually just be one that comes to you. And to your group, maybe that would be something in the new year when there's when there's a bit more time. So I did want to actually come to your meeting. It's the beginning of October. It's the 7th to the 10th, I think, right?
Yeah.
So just I would have loved to. So I'm upset that I'm missing out, because I think, as with the meeting in Austria, I think through those interactions, that's where you really learn. And they're just incredible. So I unfortunately can't make that. Yeah.
Next time, hopefully.
Next time,
Thank you.
Yeah, cool. Lovely. I wanted to ask, I mean, are you still working on the interaction between the mother and the child and the EVs? We had that discussion a while back about, one of our interests is ARVs and the trafficking of those ARVs from the mother to the child. Are you still working in that area, or not so much?
Yeah, Subhashini is actually the one that is now mainly has been working mainly in that area, and the latest paper hopefully would be, if I am the bottleneck, I haven't been
Okay.
yes, is going to come out.
Okay.
But yeah, that is the way. We haven't initiated, that work was, in my opinion, very good because it also gave us the model that we have, gave us a very good understanding of how the interaction, the earliest stages of the interaction is taking place,
Yeah.
and how it is possible to probably influence it. I had another PhD student in Sheffield that somehow used that information, and we tried to influence it and see how we can make it more or become less.
Yeah.
Since
Very
then,
interesting.
yeah, we haven't somehow put so much effort into there. At the moment, the main effort, one of the things, at least I don't know if Subhashini or the others that are here would agree with it, I think the model that we are using is a bit too complex. Okay, it is physiology, antibody. Even in your case, for example, using the umbilical cord stem cell EVs, it's a bit too complex. And if we go to find out, even for example, this protein is there or that RNA is there, it's very hard to establish where or how it is working and be able to manipulate it. But if we can go to other species like plants or even the species that are simpler, we may be able to easier understand how the model works and how we can manipulate it if we want to also understand the mechanism of action. Then at the moment, I am somehow trying to put my efforts towards that. Hopefully, if everything goes right, we are also going to have a grant that we would be able to use this money to feed EVs to different kind of animals or different part and thing, and like that, we hope that we would bring it more to the application and see what are the active factors, what would be the effect. Then I'm changing a bit the direction of the lab, and I am coming out a bit from the reproduction part. But if there would be, what you were mentioning last time was fascinating, especially with the AIDS and all the. If it comes, I am open for collaboration, cooperation. But as the main target of the lab, I want to go more towards bringing applications with EV.
Yeah. Do you work at all on bacterial EVs?
We have done it differently. Majority of the people are working on bacterial EVs and what is the effect of them. We are working on the effect of the EVs that we can get, for example, from biofluids or from the plants on bacteria. If we can use them as alternatives for antibiotics, or how that effect is there. And even if it goes to homeostasis and how that can affect the situation, microbiota situation in the digestive tract of different animals. We are thinking if we can somehow bring in like this, it can be very effective mean for as probiotics or postbiotics, and maybe sooner can come to the market. That is
that I think is, I did a study with an honor student where we were looking at, I don't remember the source of the EVs, it might have been the citrus EVs, it might have been the HEC293 EVs, but we were looking at the effect of those just in a pure turbidity assay in E. coli. And we found that just adding the EVs to the E. coli lower the turbidity. So I don't know whether the EVs interact with the outside of the E. coli and sort of make the membrane go all weird and then they die. So I think that speaks to what you're talking about there in terms of a antimicrobial effect. So I think that's I've moved away from that. As you sort of move around and sort of reposition yourself. So I'm again, similarly, more towards the plant side because there's just so much to learn there and it's fascinating. But
especially in the environment that you come from, there are a lot of traditional medicines, traditional
Exactly.
herbs, the
Yeah.
Zulus or the others that are using it.
Yeah.
And
Yeah.
that is a rich source of going and
Yeah.
further. There was recently a very large meeting in China, Guangzhou, that they had it only on the EVs from the medicinal herbs, Chinese
Oh,
medicine.
really? Chinese
Yeah.
okay. Yeah.
And
Yeah, that's exactly, that is one thing that I'm focusing on quite a lot, is
yeah,
our indigenous species that we've got here from a plant perspective, and
exactly.
what is known about them in their use in various cultures for medicinal purposes, and can we link any of that to the EVs that we extract? So it's an area that I'm very, very interested in and working with a couple of individuals. So yeah, I agree with you.
Yeah.
Harnessing our biodiversity for good.
Exactly.
Yeah.
And let's keep the contact, and hopefully, yeah, something comes out. At least we now know each other.
Yes,
If those
absolutely.
guys get in touch with me about the rhino, I will put you in contact with
That
each
would
other.
be great. That would be great. And thank you very much for the opportunity. Nice to meet you, Subhashini. And
Yeah, me too.
hope to see you in your own environment, not now doing Move, but maybe I'll just come straight to you guys and we have some chat. So
Yeah.
cool.
Thank
Nice.
Thank you
Thank
very
you.
much, Carola, yeah, for the interesting lecture and the discussion and all. So thank you very much, everyone in the audience also for joining us with the guest lecture today. So see you next time. Thank you all.
Thank you.
Bye.
Take care. Bye, everyone.